Leading a startup team? Whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report.
While most drug developers have sought to target aberrant genes or the proteins driving diseases, Syros Pharmaceuticals has developed a proprietary platform that targets the regulatory region of DNA to not only turn genes on or off, but modulate their activity as well. The company is initially focusing on cancer and monogenic diseases. We spoke to Nancy Simonian, CEO of Syros, about the company's platform technology, the opportunities in targeting regulators of genes, and why this could present a broad new approach to treating diseases.
Nancy, thanks for joining us.
Hey, Dan, it's great to be here.
We're going to discuss Syros Pharmaceuticals, its platform for developing drugs that can regulate gene expression, and its therapeutic pipeline. Perhaps you can begin with a brief overview of Syros, which was launched by a pair of venture capital firms based on the work of academic scientists into gene control regulators that have been dubbed super enhancers. What was the vision for the company?
Well, the vision for the company is to, create value for patients by creating transformative medicines in people with severe diseases, and based on our leading expertise in gene control. And we started the company up around 5 years ago based on the groundbreaking sort of biology and technology coming out of MIT and the Dana-Farber, all around the ability to understand and analyze the regulatory regions of the genome to give us insight in terms of the genes that were important in controlling disease, both in normal cells and in disease cells. So that was the original basis for the company. And now, in just a very short period of time, we have 2 programs in clinical development, 3 in preclinical, and then, additional programs behind it, both in the cancer space and in the monogenic disease space.
What makes what you're calling super enhancers attractive targets? What are super enhancers, and do they represent new therapeutic targets?
Yeah, so super enhancers are part of the regulatory genome, and the way to think about them is they're the switches that control the level of expression of genes in your genome. And the reason that they're very attractive is they're, they were enabling discovery in the field of gene regulation. Because prior to that, people didn't really understand how one got coordinated expression of a set of genes to determine sort of what a cell's function was. Because when you think about a skin cell or a muscle cell, they have identical genetic material. So if you just sequence them, they would look the same, but of course they function differently. And these enhancers and super enhancers gave us a lens into which were the most critical genes that were responsible for cell state and fate. And we built at the company, that's been a major part of our arsenal in kind of leading the approach to how to control the expression of genes that we think are important in disease.
How do you determine what super enhancer you target in a given disease? And do you target just one at a time, or do you seek to target multiple enhancers?
Yeah. So We much of our work starts in primary human disease tissue. We think that that's the most appropriate, model system for human disease. So we can profile these regulatory regions, including super enhancers, in normal cells and then in disease cells, and we can look across a disease and we can see what's different in subsets of patients. And based on that, then we determine which are the super enhancers or the genes that are controlled by the super enhancers that we may want to control for therapeutic purposes.
And are you targeting just one of these, or is there a way to target multiple regulators of the genes?
Yeah, it depends on the approach that we're taking, but in the case of cancer where, there's maybe multiple pathways that are dysregulated, we can control various components of a super enhancer that alter the level of expression of multiple genes. And of course, we think that's very important in diseases where there may be multiple dysregulated pathways like cancer. In the case of some of the monogenic diseases and the one that we're currently focused on is sickle cell disease, we're focused on an enhancer that specifically regulates a gene called hemoglobin F, where we know that if we can modulate the level of expression of that gene, that we can have therapeutic benefits. So depending on the particular disease context, we might try to control a whole set of genes, or we might try to control a single gene. But our platform really gives us the ability to do either.
And how challenging are these as therapeutic targets? Are there half-life issues with the drugs you're developing, or issues of getting them where they need to go to have a therapeutic effect?
No, our approach has been to use a small molecules to target these regulatory proteins that are part of these regulatory regions of the genome. And in some cases, they're proteins that, again, sit down right on the DNA, but they have druggable pockets. And by using small molecules, we can get through the cellular membrane and the nuclear membrane. So the therapeutic approach that we're taking, which is a small molecule approach, really enables us to target these nuclear proteins.
The lead candidates in your program are focused on cancer. Why is that? Is there something about this approach that makes cancer particularly attractive to these therapies?
And so, our platform has very broad applications. As I mentioned, we increasingly now know that the altered regulation of genes is probably one of the most important abnormalities that happens in a whole range of human diseases. If we look at a lot of complex diseases, most of the genetic variation is in these regulatory regions of the genome. So rather than having abnormal proteins, we have normal proteins, but at the wrong levels. So again, because of that, we know that we could work, the platform can work broadly. We decided early on at the company to initially focus in the cancer space, and that was because cancer is by nature a disease where there's altered regulations of multiple genes. And we thought that our approach and platform, by targeting these regulatory elements, we could control a whole set of pathways at the same time. So cancer was very attractive from that standpoint. In the cancer space, we not only focus on the cancer cells themselves, but also on the immune cells that are in the cancer. And that's important because the immune cells aren't undergoing genetic alterations like the cancer cells. They're switching from one cell state to another, and that's all based on the alterations and the changes that are occurring in these regulatory genomes. So in some ways, our platform is very well suited to understand how to switch a immunosuppressive immune cell into an immunostimulatory one that may be important in cancer in that realm. And we have a preclinical program really focused on that in the macrophage space. And then, we also more recently have been focused on monogenic diseases, where our approach is to really focus on a single gene where we want to alter the level of expression. So we have broad applications, but we are focusing right now in cancer and monogenic diseases. But we've also utilized that broad platform in the strategic partnership area. So earlier this year, we did a discovery deal with Insight in the area of myeloproliferative neoplasm, disease area that we weren't focused in, but that we could utilize our platform in the foundation that we built here to to work on a disease and provide benefit to patients in other therapeutic areas.
Your lead candidate, SY1425, is being developed for AML. What is SY1425 and how does it work?
So SY1425 is a retinoic acid receptor alpha agonist or RARA agonist. That is a nuclear hormone receptor or a transcription factor that binds to these regulatory regions of the genome. And its normal function is either to turn on or to turn off a whole program related to differentiation of cells. And what we found by using our platform, we discovered a novel proprietary biomarker that made cells, if they had that biomarker, susceptible to the differentiation effects of SY1425. And we focused our efforts with 1425 in a type of cancer called acute myelogenous leukemia and myelodysplastic syndrome, which is essentially a tumor of sort of immature cells that are kind of halted in their ability to differentiate. And so by selecting the right patients by using our biomarker, we can drive those cells into a differentiated cell state for therapeutic benefit.
Your stock took a significant hit when you reported mid-stage clinical results at ASH. The drug failed to meet the trial's endpoints as a single agent. Only one of 58 patients had a complete response. How surprising or disappointing were those findings, and do they question the approach you're taking?
No, not at all. When we initiated the trial, we were studying the drug both as a monotherapy and in combination. The initial data that we read out last year was in monotherapy, and our combination work is ongoing. I think what we demonstrated last year was really proof of principle behind the underlying findings. So first of all, we were able to show that the presence of the biomarker that we discovered correlated with the ability of these cells to differentiate in response to 1425, which was the whole underlying premise for the discovery. And then patients taking 1425 as a monotherapy, and these were relapsed refractory AML and MDS patients. We saw 43% of patients that actually had clinical activity as measured by hematologic improvements, so improvements in their red cells, platelets, and or neutrophils, as well as reductions in their blast counts, including 1 out of 23 patients that had a complete remission. And so this was really sort of proof of principle that this drug, which is a differentiation agent, again, it's not a cytotoxic drug, could drive cells into a differentiated cell state, and that we already saw clinical activity as a monotherapy, which really formed the continued investment in the combination approaches, which I mentioned had been ongoing. And so when we combine 1425 with azacitidine, which is a hypomethylating agent, we see deeper and more durable complete regressions of tumor. And we also see that tumor cells die in response to the drugs when put together, which I think is ultimately what we want to do in the clinic with these types of combinations. And we also have a combination where we're combining SY1425 with an anti-CD38 drug called daratumumab. And that's based on the discovery that 1425, when it induces differentiation of cells, it upregulates the expression of CD38. So we can take AML cells that normally are not responsive to anti-CD38 drugs and make them now responsive to 1425. So we are very excited about those two combination arms where we're currently enrolling patients and expect to have data in the fourth quarter of this year from them, those combinations.
And what is the clinical path forward, the timing when you might be able to actually move this in combination for an approval?
Well, so the drugs right now are in phase 2, in phase 2 study in combination. So the data that we will get later this year will help to inform the path forward that we take. But we're already studying those drugs in combination.
You also have a second candidate, SY1365, which is being developed for breast and ovarian cancer, with the potential to treat other cancers. What is that, and how does that work?
Yeah, so SY1365 is a selective CDK7 inhibitor. CDK7 is a transcriptional kinase that's part of the regulatory genome. It's part of the superenhancer complex, and it's important in controlling the level of expression of a whole set of genes. And what we and others have found is that it can selectively downregulate the expression of oncogenic transcription factors and anti-apoptotic proteins. And when we treat cancer cells with 1365, those cells undergo apoptotic cell death. And in our preclinical models, we can get kind of complete tumor regressions with the drug as a single agent. And so last year we started a first-in-man study in a dose escalation phase of the study, which was in all-comer solid tumors. We plan to move into the expansion phase of that study around mid this year, and that expansion phase will include patients with ovarian cancer, both as a single agent and in combination with carboplatin. And we also will be studying it in HR-positive breast cancer following frontline therapy, advanced HR-positive in combination with fulvestrant. So our approach is to focus the drug both as a monotherapy and combination in these two different tumor types, which we've shown preclinically are very sensitive to 1365.
When I think of therapies that have the potential to regulate or modulate genes, I think of antisense or RNAi. How does your approach fit into that? Are there advantages over those other approaches?
Yeah, so I think when I think about the approach that we're taking, I would the analogy I would make in the cancer space would be to thinking about anti-hormonal drugs like anti-estrogen drugs or anti-androgen drugs, which are basically also nuclear hormone receptors, where treating with hormonal therapies can turn those cancers into more chronic diseases because of the ability to modulate a whole set of important genes in that particular tumor type. So I think the advantage of the approach that we have is by targeting sort of single proteins that are part of the regulatory complex, we could control the expression of a whole set of genes that are important in the pathogenesis of that tumor. So that, I think the advantage is, we know, and for the most part in cancers, there's not one single alteration, there's multiple alterations, and the tumor cells learn to acquire super enhancers to control the expression of important genes that are driving the cancer cell state. So by targeting those regulatory regions of the genome, we could affect multiple pathways that we think are driving disease. So I think the advantage is the ability to, with a single target, to affect multiple genes and multiple pathways. And that's different than, let's say, an RNAi approach where you're really just targeting, let's say, a single gene.
As you alluded to earlier, developing therapeutics that can target the regulation of genes and not just turn them on or off, but modulate them would have potentially very broad therapeutic effects. You mentioned sickle cell disease, but where do you see the biggest opportunities down the road?
Well, I think the opportunity could be very broad. I mean, our main focus at the company is in the cancer space, as well as in the monogenic disease space. So I think, as I said, I think it could work across, a broad range of different tumor type or tumor and disease states. And, we'll look to continue to leverage our platform through strategic partnerships outside of our core areas. But, what's been so interesting is in the last several years, we are increasingly seeing that a lot of diseases, and actually a lot of neurodevelopmental diseases, and all are really a result of these genetic variations in the expression, controlling the level of expression of genes. I think there's going to be a tremendous opportunity going forward, very broadly.
Nancy Simonian, CEO of Syros Pharmaceuticals. Nancy, thanks so much for your time today.
Yeah, thank you, Dan. It was a pleasure.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.